Good afternoon. Good afternoon. Can I have your attention? I know we're at the last panel of the day, and I think this has been so far a really very productive and good conference. And we're going to get down to some sort of nuts and bolts on what are people actually doing, what's happening in industry, what kind of products can you expect. And we have chosen three very good speakers who have very interesting topics. At least to us in the science community, they're very interesting. I hope you lawyers find it as interesting too. I'm changing the orders of the speakers from what it is in your agenda, just slightly, so that it fell into line a little bit more for me. And our first speaker is going to be Dr. Irina Polejaeva. She's the Vice President of Advanced Reproductive Technologies and the Chief Scientific Officer at ViaGen, which is a company that produces animal clones, licenses and sells proprietary animal genetics, and provides technology for animal agriculture worldwide. Prior to joining ViaGen, she was head of the cell biology group at PPL Therapeutics. In that position, she produced the first cloned pigs by somatic cell nuclear transfer, a process you heard about this morning. PPL also collaborated with the Roslin Institute to produce the world-famous Dolly the sheep, whose picture you saw. Dr. Polejaeva received a doctorate in biotechnology and stem cell biology from the National Institute of Animal Science in Moscow, and she did postdoctoral work at Utah State University. Dr. Polejaeva.
Thank you very much, Deborah, for a wonderful introduction, and good afternoon. It is my pleasure to be here, and I'll be talking about application of somatic cell nuclear transfer in agriculture. I believe I'm the only speaker today who is not talking about genetic engineering and transgenic manipulations in livestock or any other animals. So that before I go into the topic, I would like to give you a very brief introduction and talk about a company that I've been working with in the last 5 years. ViaGen has been established 6 years ago. We have 50 employees, and our major interest is in gene banking or genetic preservation of specific unique genetics and commercial cloning of 3 species. We are cloning pigs.
Closer to the mic.
Sorry. We're cloning 3 different species: pigs, cattle, and horses. Our headquarters are located in Austin, Texas. We also have facilities in 2 other countries, in Lethbridge in Canada and also Querétaro, Mexico. So in the last couple of years, major focus of the company was to collect more data primarily on reproductive performance of clones. So we've done a lot of studies in collaboration with independent labs in collecting data on performance of boars or clone boars and comparing them to non-clone animals, produced more than 600 progeny of clones. And what we found that there's no difference in reproductive performance of clone animals and non-clone animals. We've also done quite a bit of work on collecting data, safety-related data on product derived from clones and their progeny. All these data were analyzed again by independent laboratories and submitted to FDA. We submitted raw data that we will not analyze them, but FDA rather have ability to analyze them independently. So that was a big focus of about 2 or 3 years of research and evaluation. Also in the last couple of years, we were doing a lot of work in producing clones of entertainment animals or performance animals, including longhorns, bucking bulls. You can see a picture of 6 clones of Panhandle Slim in the left corner. It's a very, very famous bucking bull. The interesting thing about these babies on that corner, do we have a laser pointer?
Does anybody have a laser pointer you could use?
I think I can use, oh, okay.
Dr. Wall.
It's actually a multifunctional device, so I'm learning it now. So you can see this baby here in the corner, and what was amazing about them, as soon as they were born, they really have that mean personality. So they were chasing our animal technician 2 hours after birth. So I guess it was unique to Panhandle Slim himself. So also they pick one of the cattle, which was a surrogate mother, which was probably the skittish one and didn't like to stay around human. And they would follow that one cow everywhere. So especially when visitors are coming, so they definitely had similarity at what they choose to be their mom and the attitude. So you can see picture 4 of them. We just couldn't capture all 6 of them at that moment, but it's about 6-7 months of age. So they are about 2 years old and used as a bucking stock, so for breeding at the moment. We've done quite a bit of work with horses. So I would like to tell you a story of Scamper, who is 10-time world champion in barrel racing, which is one of the rodeo disciplines, so I don't think it's very common here on the East Coast. But and Scamper is a gelding, therefore he was not able to reproduce naturally. Charmaine James, an owner of Scamper, came to ViaGen about 4 years ago, asking us to produce a genetic replica of that unique athlete. Outstanding animal and no progeny because of the result of gelding. So it's how our equine cloning program got initiated. You can see Clayton, who is a clone of Scamper. He was born about 2 years ago, and the planning for Clayton is to use for breeding. So it's one of the examples how genetic value or genetics of unique genotype, which could have been lost otherwise, we were able to bring it back to genetic pool. And when I learned more about equine performance discipline, for example, dressage, one of the Olympic discipline, and people who work with horses and know enough about horses, I believe familiar with that. Dressage is a very unique discipline, requires a lot of attention to detail, and 95% of Olympic champions or animals performing in dressage, they are geldings. There are few mares, no stallions. So what happens there? Where you go for your genetic, people in dressage bring in stallions from thoroughbreds. No idea how they will perform, no really understanding where these animals will be able to compete on that level. That is, again, opportunity to use animals, or bring them back as stallions, and use them for breeding purposes with knowledge of their performance, previous performance. So what is cloning? Cloning is simply a breeding tool. It's give us opportunity to enhance genetic improvement as any other breeding tools. If you look in the history of other assisted reproductive technologies, it definitely has long, long history. For example, artificial insemination goes all the way back to the 14th century. It's when it first was used by Arab horse breeders. You look in another list, and we talked a little bit earlier today, both Paul and Bob were giving us information about use of artificial insemination, embryo transfer, embryo splitting. They've been used quite broadly in modern agriculture. Uniqueness of cloning is in its ability to produce a genetic replica, animal very similar or genetically identical to the original donor. Therefore, there's no genetic reshuffling, and it could be advantageous in some situations, so you can preserve exactly the same genetic replica, not just eggs or sperm, but the genotype. So I believe Bob went through some details on how cloning is done, what's involved in cloning, but I will give you just a little bit additional information on that. What we need for cloning is a tissue sample. It's very tiny biopsy tissue that could be obtained from animal and sent directly to the lab. We quite often get phone calls when people say, well, I lost one of my animals and put a chunk of tissue in the freezer. There's a misunderstanding that we're working with viable cells. We need the whole cell, not just frozen DNA. And that's very crucial to get cells in the lab in a very short period of time, place them in Petri dish, expand them, and grow And then we need also eggs from the same species. What happens then in the lab? We remove genetic material from an egg, replace it with genetic material from the cell that derived from the animal that you would like to multiply, and this material delivered into a nucleated egg through fusion. We use also a step called activation, which mimics biochemically process that taking place during fertilization. So after that, we place embryos into incubator, and depending on the species, we can culture them for just a couple of hours or several days. After that, early pre-implantation embryo transfer into the recipient, which is synchronized with the embryo development. And after gestation, depending on the species, again, for cattle, it's about 280 days, you will get a genetic copy of an animal from which tissue sample has been collected. That's the process. So what are the advantages? Again, you need to remember that cloning will not improve genetic necessarily, but you can make more animals with better performing animals. Therefore, it can improve population or a herd, but it doesn't have a capacity to improve. Basically, what you're going to use for cloning is what you're going to get. Plus, you need to look for environment component. But we have, I'm sorry, press the wrong button. But we have the ability to create genetically identical copy, and therefore, through that, enhance genetic improvement and disseminate genetic faster. We can increase quality and consistency of a herd, as well as what is important. I gave you example on single animal scamper that you can recover valuable genetic, but impact of this technology could be much more tremendous in case of disease outbreak. So again, you need to have frozen tissue preserve. In addition to semen and oocytes, you can store now tissue of animals, and you can bring these genetics back to population, back to breeding population. Talked about rare breeds. There's definitely quite a few in Europe and in South America and all the rest of the countries, breeds which are very rare and a small number of animals left. These rare breeds could be preserved, or genotypes could be preserved and again brought back into population. Genetic improvement usually extremely slow process. And I want to give you just one example. Those data summarized by North Carolina State University, giving you example of selection which was based on feed efficiency. 35 years of selection. The goal was, again, that's economic trade, that's to increase efficiency of feed conversion rate. So 1972, about 3.8 pounds of feed required to produce 1 pound of pork. 35 years later, you can see there's improvement, and there's about 32% decrease in the amount of feed required to produce that protein. That's an outstanding result, but it's not only have an impact on or economic benefit to producers, there's also environmental benefit. We need 165 tons less feed to produce about 1,000 market hogs. Isn't it amazing? So what is important there? 35 years of selection, some benefits to environment and producers. We could shrink the time that requires to get the same benefit. How can we do that? If you look in this diagram, and people who are familiar with genetic improvement or just distribution of any factors, you know that distribution of parameters usually have a bell curve. So usually pig improvement company or breeders use about 20% of their best animals for breeding. So it's what they call EBV of 20, which is estimated breeding value. So basically, if you have a very good animal, but because it's just individual animal, impact of that individual outlier is so limited on the mean of population that it's really didn't make a difference whether you're going to use them in the pool or breeding schedule. With cloning, that particular outlier could be amplified, or the value or impact of that outlier could be amplified, and therefore you can see significant improvement or reduction in the time that requires to multiply specific trait. What is important to remember, again, that those traits, regardless of what you're using, you actually use that technology to improve any trait of interest. It could be economical trait to producers when you're looking for specific feed efficiency. It could be other attributes which are consumers interested. We talk about this omega-3 milk or pork, which is, I don't know whether you heard about that publication from New Zealand, that they identify one cow which had naturally very high omega-3 milk. So again, it will take probably years and years, 25, 50 years of breeding to enrich population of cattle with that particular unique high-value milk production. And that could be done much faster using the assisted reproductive technology, including cloning. So it's also, if you're looking for impact or improvement related to animal health, you can do it again with cloning. So you can shift your population in the right direction faster. That's the bottom line of use of this technology. And we talked already about mastitis and naturally resistant to mastitis animals. Also improvement in productive life. So it's in dairy situation, we know that the productive life is relatively short now, but there are some animals which perform outstandingly for 5, 6 years. There are some individual animals which can produce a lot of product and have long, healthy, productive life. You can select these animals and enrich your population with these unique genotypes faster. So how many clones? It's again, this number is estimated. It's what we believe we have. I'm talking only about agricultural application, not animals which produce through cloning for transgenic applications. We believe there's about between 500 to 600 clone cattle in the United States. About 250 clone pigs were produced, about 20 horses, and quite a lot of doses of semen were sold from cloned animals. So I don't know whether they store and for future use, but you can go online, literally use your credit card, select a bull, and buy semen from cloned animals now. Even though it's not a topic of my presentation today as far as safety data, because it will take another couple of hours because the data set is very large, but I would like just to say that FDA collected data or analyzed data for more than 400 different studies, and they cover different aspects of cloning technology. So it's stated there that data show no difference between cloned animals and control in reproductive parameters, and also milk and meat composition that was evaluated. So milk and meat chemical composition of a product derived from clone and their progeny as safe as any other food we eat every day. I would like you to keep in mind a couple of things, that clones are not genetically modified. There's nothing has been added or removed from the donor cells which were used for cloning. These animals are exactly the same as the original donor that they produced from. Also, progeny of clones are not clones. And I wanted to use this picture because you can see on that picture a litter of pigs which were derived from the same clone boar. You can see white piglets, belted piglets, spotted piglets, and they all offspring of a single clone boar. So the clone boar, he was a crossbred animal. It's why you see all this variation in offspring. But just remember, if you keep that picture in mind, you will know that clones produce offspring which are very diverse. depending on the clone. If it's a purebred animal, of course, there will be much more similarity, but if it's crossbred animal, you will see large diversity there. And cloned animals are not going to be used as food. It will be rare because technology is still expensive. And I appreciate that slide which Bob Wall showed to all of you, that micromanipulation station, which cost about $60,000, $80,000, and the personnel which required to do that procedure. It's still a fancy way to produce animals, but it has its own benefits. So therefore, clone animals are there for breeding, not for eating. And I want to also emphasize, because of the value they have, again, I said value of these animals are for breeding, we definitely spend a lot of time and attention to evaluating reproductive performance of clones. We want to make sure that the purpose we want to use this technology for is valuable, and we know that these animals can reproduce as good as non-clone animals. There are quite a few papers published on reproductive performance in different species, cattle, pigs, and goat, sheep, etc. So basically every species that clone animals reproduce have information about the offspring of clones. And if you look in all these data, you can see that clone animals have normal reproductive characteristics. Doesn't matter whether you look in their semen production or embryo production, it is normal. Conception rate, gestation rate, birth weights, etc., health of progeny of clones, they are normal. You can see a picture of that Casey, which is an animal produced, or clone, which was produced from carcass cells, and she's there with her first clone calf. Since that, she had another 2 calves, and she has a very normal reproductive life. Quite often we get questions about longevity, and clone can live long, healthy life. That's a picture of sheep, clone sheep. They were produced by PPL Therapeutics in Scotland about a year and a half after Dolly the sheep was cloned. They're still living in Scotland, and we actually purchased them from PPL. So really the reason to monitor their longevity, and that is important for us to know. And they, 1 or 2 of them approaching 10 years of age, which is very, very old for sheep. So I'm not sure whether we have sheep person. I'm not expert, but I heard that average age of sheep in the United States is 7 years. So we're doing quite well there with longevity. Is it about right? The variation probably 7 to 10, but 10, you would agree that we are pushing that envelope, that they are having a long life. So as far as performance, it's always a combination of genetics and environment. And you cannot take it away. It doesn't matter what you are viewing. But advantage there, again, if you know in what conditions that particular animal performed the best, and we know potentials or genetic potential of that animal, you can enhance performance through environment. That's a picture of one of Holstein animals, Liz. Her genetic donor, she was a champion in dairy show, and the clone was a champion in her age category. So there are plenty of evidence that clones of champions could be champions, at least phenotypically in that particular case. So in a summary, I just wanted to highlight the point that I wanted to deliver during my presentation, and cloning is simply another breeding tool, another assisted reproductive technology. It has an opportunity to provide very rapid response to changing environment. It can enhance genetic improvement. It can decrease genetic lag, and it also have opportunity to recover viable genetics in case of unexpected injury or death or major disease outbreak. And with that, I would like to thank you all for your attention, and I guess we'll address the questions after the last presenter. Thank you so much.
Thank you, Irina. We've heard several times today already about the tuna pig, the pig that produces, from which you can get omega-3 fatty acid-rich bacon. And so that it's perhaps fortuitous that our next speaker is Joseph McGonigle, who is the Vice President for External Affairs at AquaBounty Technologies, where they still have fish-produced omega-3 fatty acids. AquaBounty Technologies is a biotechnology company that focuses on health and productivity improvements in aquaculture. He served as Chief of Staff to the President of the Maine Senate and as the Executive Director of the Commission on Maine's Future. He has also served on the U.S. delegation to the North Atlantic Salmon Conservation Organization, where he helped draft international standards for control fish escapes. And he is an aquaculture advisor to the New England Fisheries Management Council. At AquaBounty, he heads an education program to help salmon farmers and others understand fish-to-fish transgene technology, which you probably should understand by now. He has a degree in agricultural economics from the University of Maine.
Thank you very much, and I'd like to thank Bill and the organizers for inviting me to this event. It's very exciting. I'm going to talk a bit today about our fish, which we believe may be the first food animal to make it through the transgenic food animal to make it through the FDA process. We don't know that for sure because we're taking a very long time doing it. We started the process back in 1996, but we only raised the money to pay for it last year, and so we're racing now to try to merge those two things. It is a little ironic, I think, that if we were to be the first with this new breeding technology to go on the market, because salmon and really fish in general, even though aquaculture may go back 3,000 years in China, as a modern farming industry, it only goes back to the late 1960s, early 1970s, and salmon broodstocks are only in their 8th or 9th generation right now. They've only been bred formally for about 40 years. In addition, the industry itself is only about that old, and so we're not talking about going back to techniques used in the 14th century, which would be nice if we could, but in addition to having the unfamiliarity of genetic engineering, we also, I think, have the unfamiliarity of aquaculture to deal with. So I'm going to talk to you just a little bit about why we're doing this and what it is we hope to do. We should start with the appetite Americans have developed over the past 35, 40 years for Atlantic salmon. 445,000 metric tons sold in the U.S. last year. Landed value, which is to say the wholesale value, of about $1.5 billion. 97% of that comes from aquaculture in Chile or Canada. There's no wild harvested Atlantic salmon sold in the United States, and there's barely any sold anywhere in the world. There's a small amount captured and sold off the coast of Norway and Ireland, a few in the rivers of Scotland, but that really certainly never reaches the United States. Last U.S. Atlantic salmon fishery was closed right down the street from my house in Maine, as a matter of fact, on the Penobscot River, and there is no large-scale ranching or supplementation going on for Atlantics like there is for Pacific salmon all up and down the West Coast, especially in Alaska, where there's about 30 or 40% of the putatively wild catch is actually reared, starts its life in hatcheries and then is released into the wild. What's going on here in salmon is something that's you're going to see increasingly with other fish as well. The natural productivity of the ocean is at its limits. It has been for nearly 20 years now. Excuse me, it's been stagnant at something in the neighborhood of 100 million tons since 1990. 70% of that is direct consumption. About 30% is what's called industrial fisheries. They're very small, rapidly reproducing pelagic stocks, things like menhaden in the United States, jack mackerel in Chile, anchoveta in Peru. These are industrial fisheries that go back to the 19th century. They used to be captured for lamp oil. Among other things, they're used in margarine. About 60% of the total catch is actually used for conventional pet food or cattle feed or pig feed. They're mixed in as part of the mix. But aquaculture doesn't have the option to substitute at this point other sources of protein, so increasingly it is absorbing that catch. Nevertheless, there's not going to be any increase in that wild catch, and so there's a major effort going on to try to develop vegetable proteins that can substitute for omega-3 rich marine proteins. The only source of new supply is aquaculture. This is a look at a salmon farm, as they're called. It's been growing by, aquaculture generally has been growing by about 10% per year over the past 40 years. The rapid growth began with Atlantic salmon in the 1970s in Norway, and the sea cage technology is something that basically made traditional sorts of fish farming scalable and possible to integrate into the commercial food system. You see this type of technology is now used for salmon, trout, for tuna, sea bass, and sea bream in Europe. To a large extent, the vast majority of aquaculture still takes place in China and Southeast Asia in traditional ways. It's a subsistence, pond-based farming technique. And there, as well as in much of the rest of the industry, the broodstock are essentially wild. They're not domesticated. They haven't been developed. And so there's not an awful lot of the types of gains that you can get from efficient agriculture, but it will happen over time. There are environmental impacts from aquaculture. To a large extent, they're manageable, but they're not always managed, and that depends on where things are grown. Laws are only good as the enforcement behind them. A couple of quick comments on environmental impacts that have been raised or concerns that have been raised with salmon aquaculture. The biggest one is wild pathogens invade the cages where they can become amplified. They can explode in their population. You can treat them. You can vaccinate them in advance, which has had a major effect in reducing the amount of antibiotics used. Antibiotic use in aquaculture is down about 98% from what it was just 20 years ago. In the worst cases, it's like foot and mouth with cattle. The pens are essentially depopulated. And then something that the industry itself has learned over time is that fallowing is critical. The more you leave your cages empty, and let them sit and let the ground recover, the benthic sediments recover, the more you're able to break the life cycle of the pathogens themselves. And so it's possible to get an integrated pest management operation going. Second major concern, and is particularly evident when you're talking about transgenics, is gene flow from escapees. One of the most important ways of controlling that is to don't grow them near salmon rivers, don't grow them in the home range, which is essentially the North Atlantic. Salmon are grown, Atlantic salmon are grown in Chile, for example, on a large scale. They're grown on the west coast of Canada and the United States on a large scale. Those things are entirely outside the home range, and there's virtually no possibility of gene flow. Fertility management is important. The industry has resisted that because it costs about 15% of their growth rate. And so as a result, for example, if you don't have to control gene flow by managing the fertility of your fish in Chile, why should you have to do it? I'm sorry, the people in the North Atlantic don't want to do it either. And then finally, as I was saying, we need a vegetable protein. So our solution is to lock this in, and I'm going very slow to start, so you're going to have to tell me if I need to speed up.
You'll get reminded by it.
Okay.
He's got 8 more minutes.
Okay, all right. I'm going to start moving very fast. basically three major features of these fish. They have much faster growth, and so they are in the water for a shorter time. They're exposed to pathogens for a shorter time, and typically the disease problems occur in the second year when they're at sea, not the first. You have a faster harvest cycle and therefore a greater opportunity to fallow on a more regular basis. And finally, as I say, the fast growth, if you double the growth of a fish, you can afford to take a 15% hit from managing their fertility. They have a much better feed conversion rate, as you saw with the pigs, 10 to 30%. They eat less food over their entire commercial lifetime, and that reduces the pressure on those pelagic stocks. And finally, if the solutions are engineered into the fish, then concerns about farm-level compliance or enforcement in some places offshore becomes a lot less of a problem because it's essentially built right in. A quick cartoon. We used a Chinook salmon coding domain, which is the purple. That has been linked to the regulatory sequences from an ocean pout. There was a single copy in the fish that we ultimately selected. It's in a 35 base pair repeat region, which is the so-called junk DNA. It's certainly biologically inactive. And there was a marker gene originally, but that was removed in the breeding process. The only thing that is expressed by this transgene is growth hormone, salmon growth hormone, at a very low level. The only novelty in the salmon genome is the pout promoter, and that's essentially just an off and on switch. And finally, the way it actually functions, the mechanism of action, so to speak, is simply to extend the growing season into the winter. This is a picture of the fish. These are siblings. We breed them, we outcross them every generation because there's been a severe bottleneck originally in selecting a single fish. These are about 18 months of age. The transgenic is obviously the top one. That's ready for harvest now. As a matter of fact, you can see right on the tip of its jaw, there's a little pink. It's beginning to think about maturing. And you'll see its jaw will change at that point. The other one has got at least another year, possibly a year and a half to go before it's harvestable. And this is, you will see that information discussed a bit more in the paper. So I won't go into that. These are essentially the major features of the phenotype. In the early development stage, there's essentially no different from non-transgenic fish. The egg number, size, egg mass, biomass, et cetera, all these are the same. Hatch timing is the same. Degree days to when they start feeding externally is the same. In the juvenile stages is where things begin to happen. They have 4 to 6-fold faster growth as juveniles, up to about 750 grams. They are very, very much more active in tanks. You can watch them zipping around the top of the water column where the conventional fish will hide on the bottom. They have an elevated metabolism, about 50%, but it increases as you put them under stress. As they have to swim, for example, against a current, they increase, they use more energy to achieve the same effect. And they do smoltify, which is they're ready to go to sea earlier than the conventional fish, but they go at the same size. They just go earlier because they're obviously growing faster. And at the adult stages, when they would be at sea, essentially they revert back to the commercial growth rate, and they're the same size as reproductive maturity. So the major differences, there's a laser, is just here. And it's a relatively short period of time of about 8 or 9 months. Quick look at their performance. This is young juveniles. And you can see the transgenics on the top, the non-transgenics on the bottom. This is over a couple of years to essentially the transgenics, again, are at harvest size, right in there. These are fish a year older but non-transgenic, and these are fish that are their siblings, like you saw in the picture. Essentially, this shows you that they're the same size. They're within the parameters of the conventional fish when they are mature. This is the effect I was talking about. Whoops. How do I go back? These are farm salmon. These aren't controls or anything. It's just regular farm salmon. And as you can see, they also have this drop in growth rate as they move from the juvenile phase into their long-term growth rate. But what's interesting is that the transgenics do exactly the same thing, but in a much more dramatic way, and they end up at exactly the same growth rate as the conventional fish. The one difference is there's a lot less phenotypic variation in these fish. When you take them out of agricultural circumstances and you don't give them high rates of feed, they don't grow any faster. These are not our fish, by the way. These are transgenic Chinooks. They're using a different construct, but we have similar data. It just isn't finished. The experimentation is ongoing right now, and so I don't have the information. But as you can see, the wild type are very consistent in their growth rate at a lot of different feed levels, but the transgenics essentially get their high performance only when they're able to take the feed that's left in the water by the non-transgenics. And then when you feed them up at 7.5%, this is how we found out they need even more than 7.5% feed, they don't quite reach that maximum growth rate. What's going on with them, as near as we can tell from a metabolic perspective, is essentially the expected response to growth hormone expression. And that includes improved lipid breakdown and faster energy deployment, enhanced protein synthesis, which is the raw material for muscle development, better mineral uptake, so they have skeletal development, higher feeding efficiency, as I said. And as I say, this is essentially the same as in wild-type fish. The only difference is there's a scale effect because these fish are growing so much faster. The cost is paid in the oxygen economy. They require a higher dissolved level of oxygen in the water to breathe, but it's still not so high that it is the sort of thing you would run into even in nature, let alone in a fish farm where the oxygen is added. They do require higher food and oxygen for routine activity. Again, I mentioned they have a higher cost of locomotion as you increase their swimming speed. They don't tend to, how do I want to say, they tend to use their energy for immediate growth rather than storing it for winter. Essentially, this is the grasshopper and the ant kind of a phenomenon. They are, if they are not fed at high levels and the phenotype doesn't express, they are no more able to dominate feeding opportunities here than our conventional fish. And they are, however, more aggressive. They have higher social standing, but that doesn't necessarily translate into better feeding opportunities. They have poorer predator avoidance, and they tend to go right in front of a brown trout or a rainbow trout to feed. Again, I'm going to leave this because I'm running out of time, but you can see this in the paper, and there is a missing reference that may be of interest to you. This is Hallam, McLean, and Fleming, who have reviewed animal welfare issues associated with fish and aquaculture. And I forgot it in my list of references, sorry. Thank you.
I hope you all are writing down any questions you might have on your blue cards or anything else you want to. And I'm hoping that when we're all done, Mr. McGonigle will explain how you know what the social standing of a salmon is.
Mm-hmm. They bite.
They bite. I get it. In other words, the higher the social status, the more they bite? Is that right? Okay.
More aggressive. They're more aggressive.
Okay. That may be true of humans, too, in a way. Our last speaker of the day is Dr. Eddie Sullivan, who is the Chief Operating Officer at HemaTech Incorporated, which is in Sioux Falls, South Dakota. HemaTech is a subsidiary of Kirin Pharma Company in Tokyo. Dr. Sullivan is a board-certified high-complexity laboratory director by the American Association of Bioanalysis. His current work includes research that is moving toward preclinical and clinical evaluation of a transgenic animal human pharmaceutical. We heard something about this earlier in the day. He's worked on various committees that focused on the ethical aspects of animal biotechnology and has participated in projects to produce a regulatory framework for this technology. He's chairman of the Animal Biotechnology Policy Committee and a member of the Food and Agriculture Section Governing Board of the Biological Industry Organization, which Barb Glenn is here representing. He has degrees from the University of Arizona and Brigham Young University and did postgraduate studies at Utah State University in embryology and cloning. Dr. Sullivan.
Thank you, Deborah. And you'll have to appreciate my challenge in being the last speaker of the day, but if you'll bear with me, we can move on with our evening activities. So today I'm going to talk a little bit about genetically engineered animal-made pharmaceuticals and a little bit about what HemaTech does. And just for a little background information, HemaTech started at the University of Massachusetts in 1998 with the idea of being able to produce a human therapeutic protein in genetically engineered cattle. Eventually, the company moved to the Midwest, and the main reason for it was because that was where our cows were. And it was much easier to move the laboratory to the cows than to move the cows to the laboratory. And so that's sort of the background behind that. We now have about 75 employees in 3 different locations. And so I'm going to tell you a little bit about that. But with regard to talking a little bit about the background of biopharmaceuticals, there's a number of biopharmaceutical medicines in development. And it's really the doses that are required that is a factor in driving a significant demand for manufacturing of therapeutic proteins. And certainly there's an immediate need to identify new sources of therapeutic proteins that can be used in the production of biopharmaceuticals. And just as an example, IVIG that's produced from human donors has a worldwide capacity of about 16 million grams and has a worldwide need of about 22 million grams. And so just within that product alone, there's about an 8 million gram shortage of what can be produced. And so that's what's really driving some of these things. Well, what are animal-made pharmaceuticals? Well, certainly it is a method of biopharmaceutical manufacturing. And so I make this statement that animals are modified through biotechnology to become factories, and I want to clarify something. I use that term when I talk to people in the general population because I need them to understand that the reason that we use the animals is because this is where the therapeutic proteins are produced. But it's the same thing for humans. If we produce a therapeutic protein in humans, they are the factories for that particular protein. Traditional animal-made pharmaceuticals have been around for a long time, in fact. We've used porcine heparin as a blood thinner, organ rejection prevention antibodies such as AtGam that are used to help for kidney transplant rejection and that sort of thing. Antiserras and antitoxins have been around for a very long time. And certainly, as I said, they do include human-made pharmaceuticals such as gonadotropins and intravenous immunoglobulin. And ingredients in other pharmaceuticals have also come from animals like gelatin and other things. And many animal-made pharmaceuticals come from food animal byproducts after slaughter. But there are some now that are being, of course, produced also in genetically engineered cell culture systems. And an example of that would be monoclonal antibodies. Well, what kind of animals are used in producing animal-made pharmaceuticals? And I certainly have a list here, but it's not exhaustive. Cattle, horses, goats, chickens, swine, sheep, rabbits. And what kind of animals, what things from the animals can we produce pharmaceuticals from? Well, certainly from blood. We know the examples of these things being produced from milk, Egg whites, certainly egg whites produce vaccines and that sort of thing, and also from semen. Well, what's the potential? Well, certainly diseases such as immune deficiency conditions, whether they're primary or acquired immune deficiencies like AIDS, cancer, perhaps anthrax, rheumatoid arthritis, or other autoimmune diseases like Crohn's disease, multiple sclerosis, psoriasis, malaria. There's a lot of potential out there for the types of diseases that can be treated from animal-made pharmaceuticals. Well, why do we do this? Well, one of the reasons is that certainly in the production of these pharmaceuticals, we can leverage some well-established agricultural methods. There's a high production efficiency, large amounts of proteins. And one example of that is antibodies themselves that I'm going to talk about HemaTech's project a little bit. A normal cow can produce up to 20 grams of antibody in 1 liter of plasma. And so it's a very efficient system for being able to produce very large amounts of these antibodies. There's also a lower production cost than traditional methods of protein development, which means that there is a reduced capital investment and lower cost to patients. And I'll give an example of that in just a second. There's production processes and potential concerns are not likely to differ all that much from current practices using animals. And what I mean by that are the difference between genetically engineered animals and non-genetically engineered animals that are used to produce pharmaceuticals. And some proteins for biopharmaceuticals can only be produced in animals. And certainly large-scale production of polyclonal antibodies, and I'll explain what that means in a minute, can only be produced in animal B-cell populations within the animals themselves. And so, is there a need for these? Well, certainly the need for human polyclonal antibodies is growing in many areas, including infectious disease, toxin therapy, cancer, organ rejection, and as I said before, both primary and acquired immune deficiencies. And so there are a number of biotech medicines in development. The doses that are required are really what's driving this. I've kind of gone through the list. Some of them that certainly are considered are viral infections, neonatal infections like ear infections and diarrhea, certainly antibiotic-resistant bacterial infections such as methicillin-resistant staph that's gotten a lot of press recently is one where this could potentially be a helpful therapeutic. And cancer, certainly in both the treatment of chemo or radiation therapy-induced immune deficiencies or in the treatment of the cancer itself. Viral infections, toxins, and also for biodefense, such as making antibodies to botulinum toxins, smallpox, or anthrax. Well, currently the only source of fully human polyclonal antibodies is human donors. And certainly the dose per treatment is high, for example, for IVIG. It could be up to 35 grams per patient per month at a cost of $3,000 to $5,000 per month for these patients. And so one of the goals of being able to produce these in animals is that we can reduce the cost to patients for these kinds of treatments. Well, how do we do this? Well, there's really two steps in our process. And the first one is to insert the human antibody genes. And I'll explain exactly how we do that in a second. And the other one is to knock out the bovine antibody genes because they really act as a contaminant in the process. And so what the eventual goal of this is to be able to produce a cow that produces pure human antibody, has high production capacity. And it's what we call a transchromosomal cow. So essentially what our production method is that we would take a genetically modified cell bank, and we use cloning as an enabling technology, and I'll explain why in a minute. We can take these cells and clone the animals. We can formulate vaccines against human diseases and vaccinate these animals, and then collect plasma, and we collect the plasma the same way that you collect plasma from humans. In fact, we use the exact same machines. And then you have a downstream process where you take the plasma and you essentially purify all of the other proteins out of the plasma and eventually end up with a purified antibody product that can be used as a human pharmaceutical. Well, what is this transchromosomal? Well, essentially, because the human antibody genes are so large, and we want the full repertoire of the human antibody genes in these animals, we essentially use what we call a human artificial chromosome that is essentially part of two human chromosomes, human chromosome 14, and then either human chromosome 2 or human chromosome 22, depending on what light chain we want on these antibodies. And so we manufacture or develop this human artificial chromosome, and then we put these into bovine cells. And then, of course, there's the issue of being able to take the cell itself and make a cow. And so we use the cloning process in order to do that. Now, I know this has been done several times, but essentially we take the genetic material out of an egg, we combine it with one of our cells that has been genetically manipulated, and create embryos, which then are transferred into cows, and 9 months later we get our calves. And the reason for doing this is because during the genetic modification process that I'm talking about, these cells have to undergo several rounds of genetic modification. And during that genetic modification of the cells really reach their maximum lifespan, and they're becoming what we call senescent or near death. And so we can use cloning to make an embryo and then make a fetus and then make a new cell line that has a new lifespan. And then we can do the next round of genetic modification and do this, and at the end of that genetic modification, they'll be at the end of their lifespan, and we just keep going through this cycle using cloning as an enabling technology until we can then produce the animal itself. And so we have done that in these animals. We have done multiple rounds of manipulation in order to knock out the cow's endogenous immunoglobulin genes. We've also introduced the human artificial chromosome to introduce the human antibody genes. We're just in process of finishing up this last production phase where we can actually bring this into a preclinical development status. As I said before, the plasma from the animals is collected in a very similar way to the way that plasma is collected from humans. In fact, we use a human machine. I don't know if some of you have been to the plasma collection bank before, but that's a Baxter machine. These animals essentially work for us about 4 hours, 3 days a month, to collect the plasma from them. It collects the plasma down here, and then the blood cells are returned to the animal. And so what are some of the advantages? Well, we can produce and raise cattle in a controlled and comfortable conditions from birth. We are regulated both by USDA and FDA because we're a biomedical application. Producing a human pharmaceutical requires us to fall under the Animal Welfare Act. AMPs are the only viable production system for recombinant human polyclonal antibodies. We can immunize and boost cattle to make highly effective, safe polyclonal antibody products, and human antibodies, it turns out, will also protect the animals from animal diseases, and we've shown this by vaccinating them and then seeing if titers are actually produced by the human antibodies against these vaccines. Well, what are the patient benefits? Well, we can make antibodies against many human infectious disease organisms. We can make antibodies against human molecules or cells. They can be used for organ rejection and autoimmune diseases. We can make very large quantities of antibodies, as I mentioned before. Scale up through clinical trials and commercial production is fairly straightforward. We still got a few hurdles to go through, but essentially by producing more animals, we can produce more product. Transchromosomal cattle are already producing human antibodies, and we expect to file our first IND with the FDA with CBER in 2009, and this is a biodefense product. And so through that process, we hope to have our first product in 2011, and that's a rather aggressive schedule, but that's kind of what we're looking at right now. I wanted to just mention that HemaTech also worked on developing cows that cannot get mad cow disease, BSE. These are 12 animals that were born, as you're probably aware, developing a human pharmaceutical in cattle when many in the pharmaceutical world are trying to get away from using bovine products because of the concern for BSE. HemaTech did a project where we actually knocked out the prion gene from these animals, and there were 12 animals that were born that we've since done a clinical trial on. And these animals essentially don't get prion diseases. Well, I want to just take 2 minutes and briefly mention a couple of things about regulating AMPs, because certainly multiple federal agencies coordinate regulation of AMPs. I did mention that USDA APHIS is involved in regulating care of animals used in research or biomedical applications. And so we're regularly inspected by USDA APHIS veterinarian. FDA CVM claims jurisdiction also over genetically engineered animals. Certainly, there are other agencies within both FDA and USDA that are concerned about regulating food safety. And the reason I bring this up is because we're actually producing a pharmaceutical in a food animal. And so it's important that we're showing that we're doing the proper things as far as disposal and that sort of thing with our animals. And also then, of course, FDA CBER in our case, but certainly Center for Drugs as well as the Center for Devices regulates biological products, their manufacture and distribution as well. And so there are a number of agencies that have oversight over what we're doing, and I will just leave it there. I appreciate your time, and I guess we can open up for questions.
Do we have any questions? Did anybody? We do have some. Are they addressed to particular people? Dr. Sullivan, Mr. McGonigle, Dr. Sullivan. Is that those that's for him too? And this one is for Joe. But there was one that wasn't directed to somebody.
Yeah, but it's transgenic animals.
Okay.
It's one of these two.
Okay, Dr. Sullivan, why don't you go first?
This question says, how do I feel about using human prisoners to provide human immunoglobulin? I guess that certainly I would have no opinion one way or the other about that I could express in this meeting, other than to say that one of the advantages of being able to use genetically engineered animals to produce a human pharmaceutical, as opposed to using humans of any kind, is that, and I say this, a little bit tongue-in-cheek, but I hope you understand what I'm saying. Humans are sewers of human disease. Yeah, no. What I mean is being able to produce a product from an animal that is certainly less risky of passing disease on than humans are is probably one advantage that we could talk about. Certainly there are other processes that we use in order to ensure that diseases are not passed from the animals to to patients. But I don't think that I would be in favor of using prisoners to produce any kind of pharmaceutical.
Yeah, let me just mention that there is just as there's a whole government structure that guides the use of animals in research, there is a similar one for the use of humans, and the equivalent of the IACUCs are IRBs, Institutional Review Boards. So that kind of a activity would have to be scrutinized very carefully. But in many ways, I think it's harder to do things with animals than with humans. I think the regulations often with animals are tougher.
Yeah.
Go ahead, I'm sorry to interrupt.
One of the next questions is a good one. It says, could introduction of a human antibody chromosome into animals increase or decrease the problem of zoonotic diseases? And I think that certainly that's a valid point. An artificial chromosome has more genes on it than just the gene that we're looking at. We've actually transferred some of the co-located genes on these chromosomes into these animals. And one of the things certainly that we have had to do is look at what these genes are, and for the ones that we certainly know their function, we want to understand what they are. I can tell you that there has been a lot of effort to evaluate that and to evaluate what the impact could potentially be on any sort of disease transfer within our animals because of having having this human artificial chromosome. I don't scientifically understand how there could be a potential for infectious disease other than perhaps some retroviruses that may have been present on the original donor tissue or something that these chromosomes came from. And so there is that potential, but these retroviruses are something that in as much as we know about them, we have to test for them, and certainly that would be how we would look at testing for any disease that may be present in these animals, much less any sort of zoonotic disease. And then the other thing, of course, is the way that pharmaceuticals are produced. There are purification methods that have to be validated and approved by the regulators in order to ensure that diseases are not being passed to the donors. The same question would essentially apply to blood transfusions, plasma donation or plasma transfers from patient to patient and those sorts of things. And so there are always concerns, and we have to keep them in mind. This might be more applicable to you. I didn't. If not, I can certainly
Let me
answer
just
part of
also say that when we finish the question and answer, which will be in just a few minutes, before you leave, sit for a few minutes because Professor Reppy has a couple of important announcements.
I will take this because this is very apropos. This question is, if a transgenic animal is FDA approved for human consumption but is then crossed, does the resulting offspring need to be reapproved because the transgenes are in a different genetic background, or are all future offspring covered by the initial approval? In point of fact, we are doing this regularly. I mean, the only genetic engineering we did on these salmon occurred back in 1998. And we breed the transgenics in each generation by crossbreeding them with conventional farmed fish, which we buy from a hatchery. The fundamental issue is whether or not the gene is stable, the transgene is stable and it's durable, whether it changes its structure, whether it moves around, whether it is effectively the same product in the progeny that it was in the parent. And FDA, in fact, has required us to determine that, exactly how durable these genes are, exactly how stable they are, and whether they remain in their location. But assuming that we can demonstrate that, then yes, the future offspring would be covered by the initial approval because that is the only genetic engineering that ever occurred. Everything else is simply conventional breeding. I probably got myself in trouble here by saying that because I know I should never try to explain what FDA wants. I'd prefer to have it explained to me. The second question is, how do transgenics compare with conventional fish with regard to carcinogens, which has been a problem with farm Pacific salmon? This is a little, it's a little off. The problem with carcinogens in farmed fish or any kind of fish has to do with things like dioxins and PCPs in the environment, and it's the feed fish or those industrial pelagic stocks that consume this, and then it gets passed up the food chain. Insofar as we are using fish meal from the Northern Hemisphere off of Europe or off of the United States, where there's a lot of these kind of pollutants in place, then the level of PCPs in the salmon flesh can rise. If we are using sources from the Southern Hemisphere, which has not been as subjected to human insult over the years, then those numbers are lower and the problem is less. I will say that even fish coming from Norway or coming from the east coast of the United States or the west coast of the United States and Canada are all safe to eat. They're all, the content of PCPs is below the FDA level. There is a very, very long-standing fight going on, not directly between EPA and FDA over whose standard will be adopted for these pollutants, but rather between the various campaigning groups who are fighting over which standard should be adopted. But essentially, they're two different standards. EPA has a standard for wild-caught fish caught by angling by recreational fishermen, and it's essentially it's higher than lower than FDA's, but that's primarily because of the expectation that these are people who are subsisting on those angled fish. So the problem here is not with salmon per se, it's with any fish that swims in polluted waters, and the problem of bioaccumulation as you move up the food chain. But again, these things have been outlawed for perhaps 2 decades, and although they are persistent, they do eventually disappear, and the content in the wild fish is decreasing. Eventually it will be gone. Finally, when do you expect a decision from FDA on the salmon? As I said, we started this in 1996, and we thought it would be 98. So, no, this is a process I think that you can target, but in the end, because it is science-based, anything we put in front of FDA ultimately has to be analyzed by them, and the first thing that could happen is they say, oh my God, there's a hole big enough to drive a truck through. Why don't you answer this question too? You know, any submission can raise a new set of questions, and so although I have made predictions in public in the past, I've learned to keep my mouth shut.
Thank you. I think these have been excellent speakers, and I hope all of you lawyers in the crowd or would-be lawyers have learned a little bit about science today. Professor Reppy.
One point to remind everybody that parking tomorrow is in the lot at the back of the law school. There will be an attendant there keeping the gate open until about 10 minutes at noon. Have a good evening.